Hi friends, welcome back to a new video in Microbiological Insights. In this video, we are going to see about the important biochemical test, that is the indole production test. And these are the subheadings we are going to discuss here in detail. So now let's see about the general introduction of this topic. You all know about the enteric bacteria. Enteric bacteria is responsible for causing the foodborne and waterborne diseases. These enteric bacteria mainly lives in the intestine of humans and other mammals. And this enteric bacteria belongs to the family Enterobacteriaceae. Most of these bacteria are short gram-negative non-sporing bacilli. And this Enterobacteriaceae can also be subdivided into lactose fermenting bacteria and non-lactose fermenting bacteria. Some of the examples of Enterobacteriaceae, which includes pathogens, are Salmonella and Shigella. These two are lactose non-fermenters. Occasional pathogens such as Klebsiella and Escherichia coli are lactose fermenters. Protease is a lactose non-fermenting bacteria, and normal intestinal microorganism is the Enterobacter species, which is a lactose fermenting bacteria. The differentiation and identification of this enteric group of bacteria can be made possible by using these four tests, that is the IMBIC test. Here the I stands for indole production test, M stands for methyl red test, V for Voges Proskaver, and C for citrate utilization test. This indole production test is one of the important qualitative biochemical tests. This test determines the ability of a microorganism which can degrade the amino acid tryptophan with the help of the enzyme tryptophanase. So, if the enzyme is secreted by the organism, the tryptophan amino acid is converted to indole, which is the end product of this test. Traditionally, this method is used to distinguish the indole positive Escherichia coli bacteria from indole negative Enterobacter and Klebsiella species. Most strains of Escherichia coli Proteus vulgaris break down the amino acid tryptophan and release indole as the end product. Now let's see about the principle of this test. The amino acid tryptophan is mainly found in all the proteins, and in the medium this indole production test is made possible by the presence of tryptophan amino acid. This tryptophan amino acid is present in casein and animal protein. So the medium sim agar, tryptophan and tryptone broth contains the amino acid tryptophan. Sim agar means sulfide indole motility agar. From the name itself, we can understand this agar is used for determining three tests. That is for determining the H2S production or hydrogen sulfide production, then indole production test, and for determining the motility of an organism. So for these three tests, the sim agar is used. The basic principle of this test is, if the bacteria secretes the enzyme tryptophanase, it can undergo deamination and hydrolyze the tryptophan amino acid. This is the structure of the tryptophan amino acid. What's the end product generated here? Indole is the end product. It is formed by the reductive deamination from tryptophan amino acid via the intermediate molecule indole pyruvic acid. So, the tryptophan is converted to indole pyruvic acid, then to pyruvic acid. If the microorganism can have the ability to synthesize or produce this enzyme tryptophanase, this tryptophanase enzyme catalyzes the deamination reaction. During that time, deamination means removal of amine group, no? So, during the deamination reaction, the amine group of tryptophan is removed. The tryptophan is converted to indole pyruvic acid, ammonium, and some energy is released. Here the coenzyme is pyridoxyl phosphate, and the indole is not used by the organisms, so it gets accumulated in the medium, but the end products pyruvic acid and ammonia is utilized by the organism to satisfy their nutritional needs. After incubation, you are going to add the COVAX reagent. The COVAX reagent, which contains hydrochloric acid, para-dimethyl aminobenzaldehyde and amyl alcohol. So what happened after the addition of COVAX reagent? The indole reacts with the COVAX reagent and it produces a quinoidal compound. It turns the color of the solution from yellow to cherry red. And the COVAX reagent contains amyl alcohol. No, this amyl alcohol is insoluble in water. Red color formed after the addition of COVAX reagent will form an oily layer at the top of the broth. So you can see the red coloration at the top of the broth. If the red color is formed after the addition of COVAX reagent, means it is positive for indole production test. If the red color is not formed after the addition of COVAX reagent, means the microorganisms does not synthesize the enzyme tryptophanase. No? So we termed as the bacteria as indole negative. And these are the biochemical reaction which occurs within the bacteria. Tryptophan, which is present in the medium, so the microorganism synthesizes the enzyme tryptophanase. The tryptophanase catalyzes the deamination reaction, so the amine group of tryptophan gets removed and it is converted to indole, pyruvic acid and ammonium. And these two are utilized by the microorganisms and indole is not utilized by the organism. So after the addition of COVAX reagent, which contains para-dimethyl aminobenzaldehyde, hydrochloric acid and amyl alcohol, the indole is converted to a quinoidal compound or rose indole dye, which produces the cherry red color, which indicates the positive result. The procedure of the test is, you can take a sterilized test tube and add 4 ml of tryptophan or tryptone broth. Then inoculate the test tube with the microorganism and then incubate for 18 to 24 hours at 37 degrees Celsius. Then after incubation, add 0.5 ml of COVAX reagent to the broth. If the red color is formed, means it indicates the positive result. Absence of ring formation indicates the negative result. We are going to see about the uses of SIM agar. Sulfide indole motility agar. We are going to detect the H2S production with this help of this medium. No? So the medium contains ferrous ammonium sulfate and sodium thiosulfate. These are the indicators of H2S production. The H2S reacts with the ferrous ammonium sulfate and forms a black precipitate of ferrous sulfide. For motility, you inoculate the organism in the SIM agar, and after incubation, you will see the motility along the stab line. The SIM agar contains tryptophan amino acid, so the bacteria secretes the enzyme tryptophanase and produce indole. The indole is also detected with the help of this SIM agar. For the determination of motility, you can stab inoculate the fresh culture into the center of the SIM agar and incubate the tubes. After incubation, you can observe the motility. The diffuse growth outward from the stab line or turbidity throughout the medium indicates the motility or the motility of the organism. And for H2S production, the media will change into black color. And to detect indole production, you are going to add 3 to 4 drops of COVAX reagent. If the red color is developed, means it indicates the positive result. No? So these are the composition of SIM agar. It contains casein, peptic digest of animal tissue, ferrous ammonium sulfate, sodium thiosulfate, agar. Tryptone broth means tryptone, sodium chloride. COVAX reagent, which contains dimethyl aminobenzaldehyde, hydrochloric acid and amyl alcohol. And observations are, positive test means I already told, no? The formation of pink to red color ring on the top of the layer, no? After the addition of COVAX reagent indicates the positive result. Some of the indole positive organisms are Escherichia coli, Aeromonas hydrophila, Haemophilus influenzae, Klebsiella oxytoca, Enterococcus faecalis and Vibrio species. And negative test is absence of cherry red color ring formation. Indole negative organisms are alkali gene species, most of the baseless species, Enterobacter, lactobacillus species, most Klebsiella species, Neisseria species, Pseudomonas, Salmonella, Serratia and Yersinia species are indole negative organisms. This is the indole positive tube. You can see their cherry red color formation here, and this is the indole negative. And this is Simagar tubes. Simagar, this is stab inoculation, no? So if the organism grow along the stab line means it indicates the motility. The organism is motile. But here you cannot see the growth of the microorganisms. Already we saw about the indole tube test method, no? Now you are going to learn about the indole spot test method. The principle of the indole spot test is the indole combines with para-diamino cinnamaldehyde in a filter paper matrix means it produces blue or blue-green color compound which indicates the positive result. So the procedure of the test is you can saturate the filter paper with several drops of indole spot reagent. Then with the help of the inoculating loop you can pick a portion of the actively grown culture and rub onto the top of the reagent saturated area and wait for 3 minutes. If blue color developed within 3 minutes means it is positive for indole production. The positive reaction is the development of blue to blue-green color within 3 minutes. Negative reaction means the development of pink color or it remains colorless. In the case of Providentia alcalifaciens, the positive reaction is the formation of red-violet color. This is indole negative, the formation of pink color. Indole positive means the formation of blue color or blue-green color. This is the spot test COVAX reagent. Para-dimethyl amino cinnamaldehyde, hydrochloric acid, deionized water.
If you like this video, please don't forget to subscribe, share, like and click the bell button. If you have any more doubts, you can write your doubts in the comment section below. Thank you.